Hi, Terry. Can you hear me? Hi, you might be muted.
I was default muted.
No, I can't hear you. So maybe it's my headset. Hang on 1 second.
Hello, can you hear me now?
Hi. Yes, I can. What is your background? Is it a curtain or a tile?
Yeah, it's a fabric thing that I have in my dining room. I'm doing this in my at my dining room table, and it's a tapestry kind of thing that I have behind
Wow, that's gorgeous. It's hard to tell if it's fabric or tile, and it matches your clothes perfectly. And your dining room tile color. No, it's gorgeous.
Oh, thank you. I decided I wanted to be out of here so I could stand up because it's hard to.
Standing is, I like talking and standing and talking. When people just casually in the room and say, oh, you can give your room sitting down, it really is hard to kind of present somehow.
It is. I need to keep, at my age, I need to keep my juices flowing here and keep my
Great. I would love to talk about anything if we have time, but I just want to make sure that we have your presentation set up and we can just make sure that it works. Do you have any videos or audio?
Say that again. So what did you say?
Do you have any videos or audios?
Yes, I do have one little video of Tim Brown, which I thought you might enjoy. So, so I did want to check that out. So what should I do? Should I just, do you want me to share my screen?
Yes. Let me, I need to make you a co-host, and then the captioner is in the room. So she's just getting ready, but I will make her the captioner, but you should be able to share your screen.
Okay. Let's see, where is it? That's weird. Where is my screen?
Ooh, do you have notes page and the screen separately, and or it doesn't matter?
Let's see if that works. Does this work?
All right, looks like it's working.
That says whiteboard though. That's not what I want.
Oh, sure. You can do an old-fashioned lecture over Zoom as well.
Yeah, but I don't know why.
There's no share screen. Usually when you go to share screen, there's a whiteboard, but there's also the other options as well.
Yeah, it's just that it's not showing me the screen, that I want to share, and I don't know why. It's funny. Now I'm going to try. I think I'm going to try. I'm going to stop sharing, and well, I'm not sharing yet. Actually, let me just try taking my auxiliary monitor out and putting it back in, because that's what I'm trying to share.
Okay. Yeah.
Just see, maybe for some reason.
I'm sure it's not the first time you're doing it. So.
What's happening? Try it again.
I apologize in advance. I'm on hotspot, personal hotspot today because the network went down yesterday, but it's working well, surprisingly.
Okay. Oh, how about this? Is that it? Try that. That's not right. Why is it doing that? I don't know. This is strange. I've never had this happen before. So let's see.
Well, I never had this happen either. Sorry.
So. Put this on. See what happens if I do this.
The whiteboard again.
This whiteboard thing, and I don't even know what that means.
I'm sorry. Let's Usually when you go to share screen, the first one is whiteboard, and then you have a bunch of screens that you're opening up and it's not showing.
Okay, I think I might have it. Let's try it.
Okay, great. Looks
Okay, that's it. Whew. Okay.
good.
It wasn't showing up. It wasn't looking like my screen. So I'm hoping that if I do this, you're seeing the right screen, right? Yep.
It's the opposite, so you might have to click on swap displays.
Okay. Yeah, I did. Thank you.
Great.
Is that it?
Excellent. Perfect.
Shall we go to the video just to see if that's going to work?
That would be great.
Okay, let's see if I can get there quickly. It doesn't want to do that. I have to go through it like this. Can't seem to get there before. Wait, there you go. All right. Let's try this. Now, shall I try just playing this?
Yes, that'd be great.
I was in the wrong place, but that's okay. How about the sound here too?
No, I don't hear the sound. Are you wearing headsets by any chance? Oh, I do hear it.
Much greater accuracy.
Okay, yeah, is there a way, let's see, for me to, I think there is something, audio settings. There's something in here. What about show in meeting option to enable original sound mute?
That's good. If you do that, I think that works well.
Let's see if that works. Let's try it again.
Has been possible with other types of biological.
Yeah, sounds good.
385
Okay.
participants.
Great.
From 3 to 20 years old.
All right, good.
Oh, that's nice. Explaining the study in a video format.
That was a current biology paper, and they asked to do a video abstract.
I see. Yep. Yep.
It's the first time that ever happened to us. So, so Tim did it, of course, because he's the lead author on it. So,
Oh, wow. That's great. That's great. I have never actually watched a video abstract before, but I do know that's a thing.
yeah, I don't know. I think maybe they put it on the website or something. I don't know.
I think so. And for social media and other things.
Yeah, well, it's great for me because I'm able to use it all the time when I
you don't have to talk for several minutes.
Describing it than I would be. So
great. Can I just talk to you about the logistics, just so that we make sure we have time, since we have about 4 minutes? Do you usually take questions at the very end and people type in the chat box? And is that your preferred style, or do you have a different style that you prefer?
I'm terrible at watching the chat when
I will watch the chat, but I don't have an admin, so I'll be kind of distracted, but I'll definitely watch the chat.
I'm very happy to take them. If you could watch the chat and tell me and actually help me.
Which one's more urgent or not?
And yeah, if you can just interrupt me and anytime, that's fine. But it's just I'm not good at watching the chat. So
yes, no, of course,
my multitasking is getting worse and worse and worse. So but also, you know, at the end as well, you know, if I'm struggling to find the chat because I've got all these screens and everything, feel free to just tell me or call out people who might want to ask me verbally.
Great. Yes, great. Thank you. Also, I want to check in how long your talk is, if you have an estimate, without interruption.
It is about, I think it is about an hour, maybe a little less than that.
Okay. I might just say, if it's okay with you, that we will take urgent, kind of burning questions and to mark them as urgent. If not, we'll take it at the end. The only reason being that some people will have their next meeting on the hour, even though we advertise it as being over an hour. So, and people want to hear most of their more talk, even though questions are usually very interesting as well. So I'm going to say if it's urgent to type urgent, and then we'll take those, but then otherwise we'll leave those to the end. Okay, so screen, I have my checklist here, so I'm going through this. Let's see, we tested all this out, and then the captioners here. So I think we're all set. Just while I have you, so we have the bulk of the kind of the main study. I mean, this is for the pling study that's been going on for years. I apologize, but we are persistent and we are getting things done with all these additional analysis and controlling, and the Lanchine score has been tricky because of it's the best data that you could do this on, but yet it's not as big as behavioral data in terms of hundreds of people. So it's a little bit tricky and finicky, but I think our bottom line that we started off with in terms of the results are still holding. And so we've been thinking back and forth. We have the basic kind of first proposal of the idea that we're presenting at the conference, but also it's the same thing as we've done before, and we shared it with you, but the conference is coming up. We'd love to have a meeting with you to get your input to see if you think it's okay, if we're missing anything, if you have other suggestions, or if you just okay it. That would be fantastic. Is it okay if we send a meeting invite to you and request a meeting?
Absolutely.
Okay.
My life is, as you can imagine, and yours
Crazy.
too, since we're both now responding to the past thing. But well, we didn't have anything else to do, right? But yes, I mean, absolutely. Just let me know, and and also, you know, if I have an absolute conflict or something with the time you have in mind, Emily Jerome manages.
Yeah, we'll keep her in the loop. Great, thank you.
She'll respond she'll make sure nothing falls through the cracks.
Great, thank you. So I want to start, if you don't mind unsharing so people can see your face and better. So I'd love to start with that if it's okay, unless you think that we'll never get the screen back and you want to keep it on, that's fine. But if you think we can get it back, it'll be wonderful if we can take it down once.
That's fine with me. You want me to stop sharing?
Yes, if you can.
Hopefully it will all work. Yes. Yes. Oops. Stop sharing. It's not stopping. I can't.
Escape.
And it is not, yep, I guess I have to do that. Okay, let's try it again. I'm in the wrong screen.
Right, stop sharing should work.
There we go.
All right, great. I'm gonna let people in. Do you have any questions?
Nope, I'm not now.
Okay, great. Thank you so much for making the time today. I know so many people are very excited, even though summer is starting for some universities already. There's a lot of great interest, so thank you so much. And I'm going to start letting people in, and hopefully my personal hotspot's going to survive this time. Let me see.
Same here. Oh my goodness. That's a lot of people.
Yes, no, there'll be a lot more. So, welcome to the BIRC Speaker Series. We're going to wait for a minute or two until I can stop hitting admit all button and it calms down, but we'll get started in a minute or two. I see a lot of repeat customers and familiar faces. It's always wonderful to see you back. All right, and I apologize in advance. Go ahead.
No, I see some very familiar faces there. I was not expecting to see, but.
It's like your big birthday party. All your good old friends are showing up.
Wow, that sounds great. We all need that, right now.
We all need that right now, yes, for sure.
Right.
I am going to do. All right. So let me share my screen. Can you see the right screen? Do you see the right screen? Yeah, okay. Is this the right screen?
I see a white screen. White.
Oh, wow. It might be doing the same thing that it did to you because it's supposed to be a presentation mode. All right, I'm not going to do this because I know we went through this with Terry earlier today, but everyone, probably many of you have seen this slide before, so I'm going to not share the screen, but just introduce our speaker and talk about our speaker series. So welcome to the University of Connecticut Brain Imaging Research Center, or BIRC Speaker Series. I know many of you have heard this over and over, so please check another email or so while I'm talking until we get to Terry's part. I am the host, Miko Haift, Director of BIRC and Professor of Psychological Sciences with secondary appointments in math, neuroscience, psychiatry, and education. Over the past 2 years, we've hosted a number of speakers, as you can see on the screen, which I usually put up, where most of the recordings are available on our website as well at birc.uconn.edu. We've had about 3,000 people joining our series from over 150 institutions all over the world. So thank you so much for helping us make this event successful. And we will continue on even post-COVID, since we started pre-COVID as well. We recently had a fantastic lineup of West Coast speakers, such as Dean Mobbs from Caltech, Kelly Grill-Spector, and Russ Poldrack from Stanford, and today Terry Jernigan from UCSD, who is the last BIRC Speaker Series for this academic year. In September, we have Marley Liu Gorno-Tampini from UCSF on aging and language processing, Alex Martin from NIH on object processing, and Mariah Thomason from NYU in October on fetal imaging. So please stay tuned and sign up to receive more information about future talks and past recordings by emailing birc@uconn.edu. As we all know, some housekeeping items that you all know by now. Please keep yourself muted, and then we will try. I usually try unmuting to clap before and after the talk, but today my network is down and I'm relying on a personal hotspot, so we will not do this. And usually, even when I do have networks with a lot of people joining, we do have a lot, there's usually a delay, so I'm not going to try to do this. If you have questions, please enter it in the chat box. If it's urgent, please mark urgent at the very beginning, as I wrote in the chat box, so that we can address those during her talk, but otherwise we'll take them at the end. So let's get started. Today it's my greatest pleasure to introduce Professor Terry Jernigan, whom you would absolutely know if you were a developmental cognitive neuroscientist or substance use researcher at any neuroimaging researcher. And if you don't, probably you should go and do a little bit more homework and reading. Terry Jernigan is Professor of Cognitive Science, Psychiatry, and Radiology, and Director for the Center for Human Development at the University of California, San Diego. She received her bachelor's degree in social sciences from UC Irvine, and then her PhD in clinical psychology from UCLA. She was staff and faculty for a short time at Palo Alto VA Medical Center at Stanford, and after that, she's been at UCSD. For over 30 years, she has studied the human brain using non-invasive imaging. She has focused on a number of different areas over time, but primarily on brain aging and neurodegenerative disorders, neuropsychiatric and substance use disorders, and more recently, or relatively recently, on brain development and neurodevelopmental disorders. For the last decade, her central research interest has been on the developing human mind and brain, with a focus on dynamic neurodevelopmental processes that give rise to human individuality, and how these processes are affected by experience, substance exposure, genetic variation, and other factors. She's co-director of the Coordinating Center for the ABC study that we all, or most of us, know, and she has served on the National Advisory Council on Drug Abuse and the Council of Councils of the National Institute of Health. Sounds very fancy. She currently serves on the NIH Helping End Addiction Long-Term, or the HEAL Initiative Working Group, and on scientific advisory boards of many research organizations in the United States and Europe, such as the Human Connectome Project. Among many honors and awards, she is the recipient of the Houghton-Locher Prize in 2014 from the Flex Society, Developmental Cognitive Neuroscience Society. So she's an exceptionally prolific researcher with over 200 peer-reviewed publications. And if you see her CV, you will see her not only as a prolific career and writer, but her deep interest in and creativity in many of the scientific areas she has touched upon. She has always struck me as someone with extreme scientific curiosity and rigor, but also a very collegial and generous researcher, always willing to share her tools and data and her expertise as well. And you will see that also if you have a chance to see her long resume, which I was studying yesterday. So it is such a great pleasure today to have Terry as our last speaker for this academic year for the BIRC Speaker Series. Please join me in welcoming Professor Terry Jernigan. Welcome. Thank you so much.
Well, thank you very much. I'm going to share my screen now, I guess, if that's okay.
Yes, hopefully it works.
Let's hope it works, right? Okay, and let's see if I can get the right screen to come up.
Yes, it works.
Is it the right one?
Yes, it is the right one. Yay.
Try starting this.
It's the right screen as well.
Okay, great. Well, thank you so much for inviting me to talk to you today in this seminar series. I've been watching the announcements of the speakers. Unfortunately, it conflicts with an ABCD, regular ABCD meeting that I usually chair. But it is such an honor for me to be among the people who are invited to talk at this seminar. I'm very much humbled by it. But thank you so much for coming to hear me today. And so what I'm going to talk about today is this relatively new paradigm named by our brilliant colleague, Thomas Paus, which he referred to as population neuroscience. Many of you probably know him. And specifically the version I and many of you probably are interested in, and that is the developmental population neuroscience paradigm. And so what is it, you might ask, and why are we doing it? And I'm going to tell the story from a personal point of view, starting with the questions that motivated my commitment to this paradigm. But let me first say that without much risk of controversy, I can probably safely assume that we all want more people in our society to thrive intellectually and creatively, and certainly fewer people to become disengaged and perhaps fall prey to damaging dependence on substances or other habits, and for more people to achieve a greater sense of well-being. But we would also all agree that some people learn more, create more, and become more emotionally resilient. And some just have more fun than the rest of us, right? My colleague, B.J. Casey, who's here today, for example. And the question is why? And for answers to this question, we could probably achieve much less consensus. Is it because people are constitutionally endowed with different personal assets, so-called native intelligence, motivation, optimism, capacity for pleasure? Or is it because of formative, sometimes transformative, experiences that either enhance or diminish their lives? Well, for the last 15 years or so, my colleagues and I have become particularly interested in this question. That is, the origins of these kinds of differences between people, which we call behavioral phenotypes. And they can be things like skills, like literacy or calculation skills. They can be levels of expertise or knowledge, more ineffable qualities like creativity, emotional or social biases. In other words, relatively persistent traits and habits. Individuality, if you like. Now, many people find questions about the origins of these differences among people intrinsically interesting. I'm sure most people here do. But from a biological perspective, the incentive to study them comes from the well-documented observation that many seriously adverse outcomes evolve disproportionately in the context of specific behavioral phenotypes. And among these are academic disengagement, for example, social dysfunction, mood disorders, even psychotic disorders, and drug and alcohol dependence. So what do we know already that constrains our models of the origins of these differences? Well, we know that most of them are moderately heritable, but the genetic architecture appears to be highly polygenic. That is, the traits may be influenced by many, many variants in the genome, each exerting only a very small effect. So what are the factors that mediate this genotype to phenotype mapping? And what accounts for the large amount of the variability that cannot be explained statistically, that is, by common genetic variation? So how do we build and test a model of a developing behavioral phenotype? Conceptually, we can begin with something like this. It's a function of the neural genotype, which we don't know what that is. It's a hypothetical, but it must exist. And in some cases, effects of environmental factors that directly affect the biology of the brain. And here I'm thinking things like prenatal or postnatal damage to the brain through toxic exposures or other forms of brain damage. But much more generally through the cumulative experiences encountering and interacting with the physical and sociocultural environment of the child. And of course, the all-important interplay between these factors, what we sometimes call gene-environment interaction, or especially gene-environment correlation. But how could we possibly achieve the integration needed to improve models of such a complex process? So many of us, some years ago, began to think, well, we really need to start by observing the developing mind and brain closely and continuously. So here at UCSD, a major first step in this direction was a project called PING, or Pediatric Imaging, Neurocognition, and Genetic. It was funded by the American Recovery and Reinvestment Act, addressing the last Great Recession. Hopefully many of you won't even remember that, but some of us do. Which was organized, and this was a project that was organized with Anders Dale and many others here in La Jolla, but also many other leading investigators across 10 other distinguished institutions. BJ Casey's one, I saw her here today. And the project had only one specific aim, and that was to create a public pediatric imaging genomics database. There was none. There were adult imaging genomics databases, and especially in older adults, but none in this age range. So we recruited participants in the missing age range between ages 3 and 20. We enrolled 1,700 folks, families and children, and had key deliverables that were QC'd acceptable in over 1,200 of them. And our key deliverables were very modest because this was a stimulus package grant, and we were supposed to do this all in 2 years. And because of this amazing team that you see here in these boxes, we were able to do it pretty much in 2 years. I think it took us 3. We had demographic, neuromedical history, and family history data. We had the just then minted NIH cognition toolbox to use for cognitive assessment. We did get social emotional phenotypes in a subset of them through our participation in another stimulus package grant, the Phoenix Rising project. And we had multimodal imaging, T1, T2, Hardy for diffusion imaging, resting state fMRI, and genome-wide genotyping, of course. Okay, now I'm just going to talk a little bit about a few observations that we were able to make. This is, maybe a kind of a boring one, but we were able to conclusively show with these two kind of primary global cortical phenotypes that the total surface area of the cortex does continue to expand continuously until about age 9 or 10, and then significantly, very subtly, but significantly contracts after that, pretty much continuously after that. In contrast, the apparent cortical thickness, that's what we measure as cortical thickness, is the average across the whole cortex of the difference between these this green and blue lines here shown in the free surface segmentation. This apparent cortical thickness phenotype that we measure declines continuously and fairly linearly across the whole age range. So there's no increase in cortical thickness, at least in this age range from 3 to 20. Anything that people have reported in the past is that really was a result of not completely separating this independent surface area increase from the thickness decrease. But of course, because we had this fairly large sample size, we were able to actually estimate these effects at the vertex level. And so we were able to produce these figures shown here, which is just showing you that this is a kind of a parameter that is the estimate of the annualized rate of change of the surface area at every vertex on the tessellated surface of the cortex. And you can see here that at ages 4 and 6, at every vertex, you can see that the increases are all in the warm colors here, that at every point on the vertex, the surface area is increasing in the yellow areas in excess of 3.5% a year. And then it slows down a little bit. So we have a different scale over here for these ages. But even at 8, you can still see that the cortex the surface area is increasing at every vertex on the surface. In these yellow areas, an increase in excess of 2% a year. But at age 10, it's a little hard to tell what's going on. All these effects are very close to zero here. It looks like there's some heterochronicity in this when the surface area peaks here. But you can't really tell what's happening between 9 and 11 because, well, they're very small effects and they're peaking. But you can see over here in 12, 16, and 20 that the surface area is contracting. That's the blue areas, to a very small degree, but continuously across this age range. Okay, and I'd like to, let's see if I can advance my slide here. Can't get over there. There. Okay, so this is one other highlight that I wanted to talk about from the Ping study. This is a current biology paper where we looked at how we could combine the imaging phenotypes to predict the age of the individuals in our sample. And I want Tim Brown to tell you about it. So I'm gonna try to hoping you can hear it.
First, we show that structural MRI scans of the brain can be used to predict a child's age with much greater accuracy than has been possible with other types of biological measures. In 885 participants from 3 to 20 years old, using a combination of brain measures from MRI scans, we predicted everyone's age within about a year on average, explaining more than 92% of the variance. What this means is that we've uncovered a developmental clock of sorts within the brain, a biological signature of maturation that accurately reflects age differences regardless of other kinds of differences that exist across people. This essentially means to measure where a person lies along a continuum of biological development made up of periods of different kinds of growth and changes over time, which you might call phases.
So one thing I loved about this paper was that we could actually kind of look under the hood a little bit. It's very much a data-driven kind of machine learning type of paper in many ways, but we could look under the hood at these different cortical phenotypes. And if I can get my cursor over there, I can show you that this is just showing you for different sets of imaging phenotypes how much age information was present in them. And so we see how the development of the biological development of the brain is sort of cascading through our different imaging phenotypes at different points in time. So early on, these are actually just signal values in the deep subcortical gray matter structures. And early on, they give you a lot of information about the age, probably due to mineralization of the basal ganglia and other deep subcortical structures. But then they begin to be less informative about age, and you begin to see these and more information coming from this. This is the diffusivity of the fiber tracts. And also you see this increasing importance of the subcortical volumes, interestingly, in the older age ranges. So it gave us some kind of clues about how to use our imaging phenotypes to look at the continuing biological development of the brain, and some clues about what might be going on as well. Anyway, so this is just one highlight from Ping. I want to quickly just summarize, though, that
Harry, can I just ask a quick question? This is not an urgent question, but it seems like it's not a good question for the end. It's a very quick one. From the one before Tim Brown's study, the question was, is the surface area accounting for the intercortical cranial volume, ICV? I think it was at the slide before. Is the surface area accounting for ICV?
Well, the surface area is highly correlated with the volume of the brain. It's not exactly the same, but the surface area is very highly correlated with the volume of the brain, and the volume of the brain is an important contributor to ICV because the ICV is the intracranial volume, and that grows in response to the growth of the brain, and so they are also highly correlated with each other. I'm not sure if that's what you're asking, but that's the relationship between them. And you
I guess
know,
if they regressed out in the analysis, if you remember, I know it's a while ago.
Oh, well, no, what I was trying to show you there was the expansion of the surface area. And so we're not regressing out. In that case, we're really just trying to, well, yes, okay. So we're trying to figure out relative to the overall cranial volume. I don't think we used ICD. Well, we may have used ICD, it's possible. Yeah, it's possible. Yes, so we're really looking at, no, this surface area effect is independent of the variability in ICD. That's true.
Okay, this person is restating the question. Are the local rates of changes in surface area normalized by ICB to enable the intersubject comparisons? But I think you got to that point. You mentioned this. Okay, so just another quick question. Did you look at gyrification? And this is from Mary Louisa Mandeli from UCSF.
We didn't look at gyrification. We didn't look at gyrification in this. We didn't use gyrification as a set of phenotypes in this analysis. No, we didn't. Though, as you probably know, there are estimates from the free surfer suite that you can look at sulcal depth and gyrification to some degree, but we didn't use it in this analysis.
Great, thank you. And please continue.
Okay, so Ping obviously has contributed to more powerful, at least at that time, data-driven analyses of genetic imaging and behavioral measures to model cognitive and brain development. There are certainly over 100 publications. It's kind of an old slide. I think it's quite a lot more than that now, because the data set is publicly available through the NIMH Data Archive. But the cross-sectional design and the very limited assessments that we collected limited the power of the study to explain developing individual differences. And the cohort characteristics really did not closely resemble the population. People were scrambling to find 3 to 20-year-old individuals, and they was not really an epidemiologically driven kind of cohort. But it did help lay the groundwork for the longitudinal multi-site study ABCD, which is what I'm going to talk about now. As I've just shown you, and as you know, of course, neuroimaging reveals ongoing biological development of the human brain throughout childhood and into early adulthood. But, you know, folks have been recognizing for quite a while that adolescence is a really pivotal time in the lives of young people under the best circumstances, associated with a lot of increasing knowledge and skill, sort of blooming of creativity, engagement in life, independence, resilience. But it's also the time when the earliest signs of serious mental disorders sometimes appear. And there's unequivocally an increase in risk-taking during this period for probably very positive reasons. Many, you know, it's a great time to get out of your nuclear family and out into the world and connect with your peers and find your partners and all that kind of stuff. So a little bit of increasing risk-taking has probably been favored in evolution, but I'll leave that to BJ to explain more about that. You know, at the time of ABCD, I think we in the extramural community and also the leaders at the NIH recognized that health and mental health and social and academic outcomes had all been associated with genetic, environmental, and behavioral factors. But in order for us to use this information for more effective preventative interventions, it seemed that we would depend upon better models of the roles of these factors to each other. Unfortunately, the limited scope, size, and method sensitivity in some cases of previous studies had reduced the impact of the work at that time. So the idea for a large-scale study of the adolescent brain originated at NIDA and was developed within CRAN, which is the sort of NIH Collaborative Research on Addiction entity. It's a collaboration between NIDA, NIAAA, and NCI. But as the planning proceeded for ABCD, and even after we started with ABCD, many other federal collaborators became interested in the study and things that they might be able to do, answer questions they might be able to answer. So now we are supported by 10 institutes and centers of the National Institutes of Health and 4 additional federal partners, NSF, CDC, National Institute of Justice, and the National Endowment for the Arts. So here are the basic aims of ABCD: to develop national standards for normal brain development. We do measure individual developmental trajectories for many outcome measures, brain trajectories, cognitive and emotional development, academic progress, for example. And we identify factors that may influence them or try to protectively or adversely. We are examining the roles of genetic versus environmental factors and their interplay, and I'll talk more about that in a minute. We're studying effects of health, physical activity, and sleep as well on brain development and other outcomes. And we will be, because of the large sample size, I'll tell you about in a minute, be looking at the onset and progression of mental disorders in some young people. And we'll be looking closely at the variability that we see in the course and severity of these disorders and relationship between mental disorders and substance use. And back to where we began, we will determine in detail how substance use affects developmental outcomes and vice versa. Okay, this is the consortium, and these are all the many people that I work together with every day. And as you can see, there's a coordinating center and a data analysis and informatics center in La Jolla. And there are 21 sites around the country, and they're just stunning people. There's 200 people in our investigator group, and they're amazing. I'll show you the picture at the end. But this is it's very important to know, this is a very highly collaborative project, not just among all the scientists who are involved in ABCD, but also we're working very closely. This is a set of cooperative agreements. They're all cooperative agreements in terms of the funding award mechanism. And we work right alongside federal investigators at many of these institutes. We have a fantastic project director, Guy Dowling, who works with us very closely, who's the project director at NIDA. And we really, I can tell you it's wonderful. And you might say, oh, well, how would that be to be working right alongside your sponsors all the time? But it's extremely helpful. It's been, I think, a major strength to our study. Okay, so here's the national scope of ABCD. So you can see we're spread out. And this is I'm going to talk to you about the cohort. So we enrolled 11,880 kids, all between the ages of 9 and 10. And it is a cohort that our enrollment was driven by epidemiological consultants. And so it largely matches on demographics, the target set for us. There are 2 sort of somewhat separate populations. The general population, what I call the general population cohort, is 10,150 kids and their caregivers. And this is what you see here in this dial is kind of the match between what our goals were for enrollment and what we actually achieved in each of these groups that were just as kind of a shorthand of the epidemiological targets that we were working toward as we were enrolling. And you can see that we did a pretty good job. I mean, it's not perfect, but we did a pretty good job. And we have good representation for each of these groups. And we also have embedded within the study a twin registry cohort. And so there are about 1,730 twins. We actually have a few triplets and things like that as well. And again, these are mostly from 4 twin sites, very experienced twin sites that have been studying twins for a long time. And they're mostly in the Midwest, and their demographic is a little bit less diverse. And so their targets were a little bit different, but they also worked very hard to have diverse representation in our twin cohort. Okay, the visit schedule is that we see the kids for comprehensive assessments with all of our kind of domains measured and multimodal neuroimaging at baseline and biennially, that is every 2 years afterwards. We do see them face-to-face at the interim years. So every year we see them face-to-face under normal circumstances. And then we have more frequent phone or web assessments. This is just an overview of the protocol from the baseline. You can see the protocols on the website. They're all presented like this on the scientists page if you go to the ABCD website. But this is just really to remind you that we have lots of different domains: physical health, biospecimens, mental health, neurocognition, culture and environment, substance use, and we have a lot of other data sources that we link external data to the kids through geolocation and other ways of linking environmental factors to them. But I just want to point out that we do have this multimodal brain imaging with structural MRI and functional MRI. We have 3 task fMRI paradigms, the monetary incentive delay, the stop signal task, and the emotional inback, which BJ gave to us and is a very powerful functional imaging paradigm for us. And resting state, of course, fMRI. And then, of course, in the beginning especially, we get a lot of information from the parents. What you just saw was the youth paradigm. This is stuff we get from the parents. But again, physical health, mental health, substance use, culture and environment. I have to highlight a few of the very important things that my colleague Anders Dale is in charge of over at the Data Informatics and Resource Center. We use an MRI protocol, which Anders refers to as a harmonized human connectome protocol. He worked with all of the vendors, Siemens, GE, and Philips, because we have these scanners at some of our sites, all of them, to ensure support for the ABCD protocols, and they are providing support for the ABCD protocols. All the vendors are. We employ fully automated computational workflows. Of course, the DARC also manages all the electronic data capture, which is central, and public sharing of all the raw and derived data from all domains, along with many of the computational tools through the NIMH data archive. And just a few enhancements to the imaging in ABCD from some of the studies that came before, certainly from Ping and so on, was we do use real-time motion correction, detection and correction which has been adapted for these multi-band protocols that the Human Connectome developed, and for functional and structural imaging, both functional and structural imaging, and distortion correction of diffusion and functional imaging for better alignment with the anatomical images. We use a relatively novel multiple b-value multi-direction restriction spectrum imaging protocol for diffusion in ABCD, which is nice because it helps us to separate the shape-dependent intra and extracellular components a little better of the diffusion signal. And that's turning out to be very helpful. And of course, the other thing that the DARC is responsible for is to do their best to do cross-vendor, cross-site calibration of imaging data in all these modalities. Now, I'm just going to say that I can't talk too much about the imaging, except to say that we have over 20, seems always to be growing, imaging biomarkers that are measured within each structure, cortical region, and fiber tract that you see illustrated here in this ROI maps. And so there are tens of thousands of variables for imaging variables that are tabulated variables that are in the ABCD data resource at the National Institute of Mental Health Data Archive. But of course, the images themselves are also available, pre-processed images, if you like, raw images, and so on. So this is a really important thing about ABCD. It has been conducted as a very aggressive open science study from the beginning. We do share the raw imaging data on fast track, so it comes out pretty quickly, just with very, very basic QC for protocol compliance. And then we do annually, we release curated data, again through NIMH Data Archive. And the most recent one was 2020, which did have at least several thousand of the 2-year follow-up visits, which means longitudinal imaging for the first time. Okay, another thing that is we're proud of, and this is really, we're proud of Wes Thompson and Halka Barch and the people who've been most involved in this. Chun Fan now is working on it a lot. The Data Exploration and Analysis Portal, or the DEEP, which is a cloud-based interactive tool for analyzing ABCD data using state-of-the-art mixed models with many factors pre-coded to facilitate nested multi-level modeling of the data, which is necessary with such a complex cohort composition. So it capitalizes on and accounts for things like race, ethnicity, SES, site, scanner relatedness, because we have a lot of relatedness in ABCD. It provides model diagnostics and advanced plotting options for users. It facilitates hypothesis pre-registration if you want to do that. And it allows expert R scripting to extend the functionality of the standard features so that the R script is right there for the model and you can change it. It offers users the option to save and download an entire self-documented analysis with the R scripts and all the associated model parameters, results, and graphics associated with them. So it's a very powerful tool, and it's instantiated in the cloud and at Enda. So you get access through Enda if you are an authenticated user there. Okay, so this is just a joke because really what I'm going to talk about now is to talk about some very early findings, just to give you a hint of what it's like, you know, what some of the ABCD results are like. There's a lot of stuff coming out, and I certainly can't tell you much about them all. But just to point out that, you know, the obvious thing here is that this is there's a lot of power in your analyses in ABCD. And I just wanted to remind you of this panel in the surface area effects at the vertex level at the 10-year-old age range in Ping, where we had, you know, 1,300 or so kids. And you can see that although it looks like maybe some areas are still expanding and some may be almost contracting already, you really can't tell because there's not enough precision statistically to tell the difference across these very small effects. But this is exactly that same age range, 9 to 11, which happens to be what our baseline age range was. And if you look at this in ABCD, you actually, these are the standardized betas, and I can tell you these are very highly significant increases here that you can see in the yellow areas, at the same time that there are highly significant decreases in the blue areas that you see here. And so here we can show absolutely unequivocally that different parts of the cortex are peaking in surface area at different times with this added precision. And it's just a snapshot to show how these very small standardized betas that you see mapped here are all very highly significant, and that just underscores the precision that you have with the imaging. So I'm going to take this prerogative to talk a little bit about some work that I've been working on with my wonderful postdoc, Claire Palmer, who is pictured here. This is the focus of our own ABCD imaging group here at UCSD, and it follows up on an interest that I've been pursuing for a number of years in the relationship between individual differences and regionalization of the cortex, that is the pattern of relative surface area in different functional areas, and individual difference variability in cognitive and intellectual phenotypes. And as I mentioned, this is Claire Palmer's work, and she actually presented these results at the Human Brain Imaging Pacific Rim Conference a year or so ago, and it was the prize-winning postdoctoral presentation. So I just have to point that out. It's now in press at Cerebral Cortex. So what we saw here, and I know that these are very hard, the maps are very hard to see, was just that we looked at both fluid intelligence and crystallized intelligence. These are just indices that you get with the NIH Cognition Toolbox. They're just really, the crystallized intelligence is really just vocabulary and reading, oral reading. And the fluid intelligence index from this is really the sort of a weighted sum of the, actually maybe just be a straight sum of the average of the other tasks, which are more what you think of as kind of working memory, more fluid intelligence kinds of things, but a variety of different tasks in the NIH toolbox. Anyway, these are the two indices that are computed to estimate fluid and crystallized intelligence from the NIH toolbox. And both of them were predicted by a pattern of cortical regionalization. So Claire used a novel method for looking at these effects, which that allows us to estimate the degree of association between a whole set of distributed effects, in this case across the cortex, and an outcome variable of interest. And here the pattern of regionalization of the surface area of the cortex is related to these two constructs from the toolbox. And the primary analysis, so look, we had two questions on this. The first question was, does this new measure provide more power than conventional voxel-wise methods for detecting a relationship between brain and behavioral phenotypes when the effects are expected to be widely distributed in the brain rather than highly focal? as we expected these to be, because we know something about the regionalization of the cortex, and we know that it is a very graded phenomenon, that we know about the molecular, the sort of the molecular distribution of those gene products that regionalize the cortex, and we know that they're very graded across the cortex. So we thought that this was an example where the mass univariate searching for localized effects approach is probably not the most appropriate. And so we applied this new method to see if we had more power to detect patterns like this. And the answer to that question was yes, we do have more power for that. And that's a long story, so I won't be able to talk about it here, but you'll be able to read it in the paper. But anyway, both of these were predicted by a pattern of cortical regionalization. And if I can get the slides to advance. But it's clear that patterns associated with these two closely related phenotypes are distinct. As you can see here, this is the fluid score, and this is the crystallized score of relative cortical surface area. And you can see that these two patterns are quite distinct from each other. And this is just showing that the difference between them. So the difference between the effects, and you can see here that the differences are fairly pronounced. This is where you can see the most striking differences by looking at this one. And this just shows that if you look at these, if you look at the fluid intelligence score, the pattern across the cortex, and then you look at what happens if you control for the crystallized, it's almost exactly the same, even if you control for the crystallized. And this is the difference between them, which is virtually none. And the same goes with crystallized. If you look at the crystallized pattern and then you look at the crystallized pattern residualizing it first for the fluid intelligence score, it looks very much the same, and there's no difference between these. So just to show that there really is a very distinct pattern of regionalization associated with these different phenotypes. And that's what I just told you. So in fact, if you look at every subtest in the NIH toolbox, you see that each one is actually associated with a distinct pattern of cortical regionalization. What does this mean? Well, what we're showing here is that the cortex of the best readers is not regionalized in exactly the same way as the cortex of the best cognitive set shifters, or even the best encoders of the story sequence, which in shorthand to me means different minds have somewhat different brains. May be obvious, but actually not too much clearly demonstrated. So why do we think these observations are important? Well, the results are consistent with a recent model of intellectual phenotypes that challenges conventional theories that a latent causal factor G underlies variation in intelligence. A model referred to as a mutualism model explains the psychometric hallmarks of G as a consequence of functional interactions between relatively distinct neural processing systems during development. And we've been very interested in this model and have been trying to sort of follow it through to its logical conclusions. And if you complement this work with other work in our group examining the heritability of these phenotypes, which I don't have time to talk about, these results underscore the importance of determining how learning experiences of children with different neural phenotypes may serve to amplify or suppress these positive interactions during development. So I want to talk briefly before I stop, if I can, about what is missing in ABCD. And of course, we've known from the beginning that what was missing was the beginning. But I want to say first that help is on the way. The NIH has decided to conduct a similar large-scale study starting in the second trimester of pregnancy for the mothers. And there's going to be a large-scale study of the very early development of the brain that kind of fills in the gap from birth, or from before birth actually, to age 10. And then that's going to be happening pretty soon, I think. So that helps on the way, as I said, on that one. But while the ABCD assessment protocol includes child and parent reports and passive measures of activities, environments, experiences, a lot of stuff we're studying, I am still concerned that we are missing the experiences of children in the environments where they spend the majority of their waking hours on most days, and that is in their classrooms. So we are not studying to the extent that we should, what's happening to kids in classrooms. And this is on my bucket list, as Fumiko knows well. So for the measurement of many phenotypes, ABCD does rely on self-report measures from parent and youth. So we need less intrusive, more robust phenotyping methods, more passive ones if possible, with higher temporal density of sampling. And although many people are excited about these new techniques for phenotyping, you know, we need for them to move along faster to be applicable to youth and well developed for these large scale studies. So I think that will help in the future. And it's very important for us all to remember that ABCD is just an observational longitudinal study, and no matter how fancy our statistics are, we cannot determine causality with it. And it will have to be these the factors that are implicated in these studies. If we think they're causal and we think they're real targets for intervention, we're going to have to follow up some kind of intervention study to prove that they are really the causal factors, because this is observational data. Okay, I have to close with a word about replicability. This is very important for us to remember, and it's important for understanding what you see coming out of ABCD. So this is a slide that I borrowed from Wes Thompson, who's our chief statistician at ABCD and a very treasured colleague of mine who's very wise, and he defines replicability as effects are similar if they're estimated in comparable independent samples. So if they are similar across different independent samples, they're replicable. And the broadly based sampling frame and large size of ABCD directly impacts the low replication rates that afflict neuroscience research. And this is just showing you kind of the sampling distribution for, so this is just a simulation where you have a true effect of 0.10, a row of 0.10 in an association. And this is just showing you, this says log 10 of the sample size down here, and pointing out that this is kind of the sampling distribution that you get, you know, when you have small studies, and this is with somewhat larger, slightly larger, and so on studies, these sampling distributions here of the effect that you observe. And you can see here that most of these smaller studies are not really powered to estimate this 0.10 sample effect. And when you have reports that are published from these small studies, they're almost certainly inflated effect sizes that by chance someone got, even though the small effect is only actually 0.1, right? So this is a well understood problem now, and I'm sure most people here already know about it, that this means that our literature is biased. Because so many studies were underpowered to estimate accurately small studies. You can see ABCD, in contrast, is very well powered to estimate this small association of 0.10, and it's significantly, the confidence intervals are very small, and but it's clearly not including 0. So anyway, this is you know, this graph shows that. Most of the published values from small studies are dramatically inflated. This is called the winner's curse. Nobody's cheating. But there have been few failures to replicate these. There will be very few failures to replicate these inflated effects, or replications with smaller observed effects that will be published. That's our publication bias, right? We don't publish the failures to replicate. If we had published all the failures to replicate of these findings, we would have figured out quite quickly that they were inflated, but we didn't. We don't do it that way. And so the neuroimaging literature is biased toward inflated effect sizes, and ABCD size studies have the power to detect small true effects. So what does this mean? It means that we should expect to see some major scientific market corrections. If so, we're going to have to, and that means many observations from ABCD will force corrections. The sizes of the effects are likely to be revised downwards. Some may be shown to be absent or trivially small. And it is important to note this scenario resembles the period in human genetics when GWAS failed to replicate many candidate gene studies, and it probably has many of the same causes. So I think it's important that we not despair and you know, we avoid the initial confusion that was caused in GWAS by those investigators searching for the genes that endow heritable traits. And we take solace from the rebirth and excitement that the field now enjoys as it gets on with challenges associated with polygenicity and the complex interplay between genetic variation and environmental factors. So I think most people who are following the human genetics field know that we are through the existential crisis that occurred right after GWAS, and people are really excited about what the actual data show us about the nature of human genetics. And in particular for our favorite phenotypes like imaging phenotypes and behavioral phenotypes. So what's next? We need to learn to construct models to describe the additive effects of many factors on outcomes of interest, kind of like the people in genetics did. And this is just my little shorthand for how I think about this. You know, we need to think of our outcomes as being a kind of model of many different effects. There will be genetic variance. There will be environmental factors, and some of those environmental factors will be correlated with genetics, and some will be uncorrelated with genetics, and then there will be true G by E interactions. And we can do this by using much larger data sets and also from combining resources from large cohort studies to fill these gaps, going to the UK Biobank for some answers and applying those to data sets like the ABCD study. And of course, there will be other large studies as well. And then of course, we have to test for causal effects with intervention studies based on reliable effects of malleable factors on these important outcomes from observational studies. So if there are no truly independent, reliable effects of malleable factors in these models, it's probably not worth going to do an intervention study because they appear to be mediated by entirely different variables. So anyway, in conclusion, let me say that developmental population neuroscience is a new, highly collaborative way to do human developmental science. Many potentially important factors that may influence the development of the child can be measured in real time. The effects of genetic, environmental, and behavioral factors and their interactions on important outcomes can be estimated. We hope that promising targets for new, more personalized interventions to prevent or ameliorate adverse outcomes may emerge. And open science will definitely accelerate progress and increase the impact of our work. And this is just the last time, one of the last times that the ABCD consortium was able to get together here in La Jolla at the Estancia Hotel. And we're really looking forward to doing that again soon, as you can imagine. So I'll stop there.
Great, thank you very much, Terry. That was a tour de force of going through the history of PING and ABCD and the future. Thank you. There's a lot of comments here, and people enjoyed your talk, but there's one question, and please do feel free to type in a question, or we can have you just unmute yourself in the future. Shalila Haas asked, the neuroimaging interval for ABCD is every 2 years. How is this interval decided?
I'm going to stop sharing so I can look at people, if that's okay.
That's great.
Okay, I also last part of that. The interval, the neuroimaging, is that what the question was?
Why is it 2 years, or how did you determine 2 years was a good time?
Well, first of all, let me say that in ABCD, we are organized into work groups. And we try to take, of all the expertise we have in the consortium, we try to have work groups that work on each piece of the design, each piece of the protocol, that are the people who have the most expertise in that area and who are the most committed to working together on, on those different parts of the brain. So that, probably the short answer is probably that our, enrollment design group, work group, came up with it. But I can tell you that part of it was expense. It's one of the most expensive things we do in ABCD. And so we wanted we were expected from the outset to do at least a 10-year study. I think everybody realized, we're stepping on this is new territory for spending this much on a single study. And it was a compromise, I think, partly, but also because people felt that it was often enough to really chart the trajectories in such a large cohort. And so it was, yeah, I think it was, a kind of a cost-benefit analysis that ended up at biennial.
Great, thank you. Other questions? People are great, thank you. So I have a question, if that's okay with you. I know in the next stage you talked about some better modeling approaches for genetic and kind of environmental factors and how they correlate. So gene environment correlations is a kind of a hot topic, and people are doing these kind of things. We're starting to think about this in the brain imaging field as well. But can you remind me, do you have twin data also? Do you have parent offspring data? What are your ways of kind of actually modeling these kind of gene behavior, gene environment correlations? Or maybe you said this and maybe I was distracted with the chat box as well. I apologize if so, but.
No, this is a really good question. It's really it's really very challenging, particularly for the gene environment correlations, right? Because I mean, by definition, they're confounded. So it's tricky. We do have a twin study. We have an embedded twin study. So, a large sample of same-sex monozygotic and dizygotic twins who are available. The data are available for modeling those. We also have a lot of siblings in the study, as a matter of fact. And
do you know the rough sample size? I'm sorry.
Sorry.
The rough sample size of these twins and monozygotic, dizygotic, and twins. I remember seeing those, but I can't remember the sample size.
That was 1,730. I think that was the twins. I'd have to go back and check for sure if that's just the twins. Or there's a few sets of triplets in there, but yeah, I think it's 1,730 twin pairs that were recruited from. Actually, there are probably more than that. I think those are the ones who we recruited from twin registries at the twin sites, but we did not exclude twins from the other side. So we actually have some twins that are just, kind of opportunistic twins that were recruited through our schools. We recruited primarily the general population samples through schools. And we did take siblings if they fell, during our enrollment period, if the kids if the siblings fell within the right age range. So if we recruited somebody at 10, and then within the enrollment window, their younger sibling became 9, we recruited them. So we have quite a lot of siblings as well. So we have a lot of family, sort of like genetic epidemiology data in there for those who like to model gene effects that way. And then of course we have genome-wide genotyping for essentially the whole cohort, all but about a few hundred of them. So, so those are ways to use conventional behavioral genetics or, quantitative genetics, statistical genetics approaches to look at these things. Now, the question of differentiating gene environment, correlated gene environment factors, that's tricky. We have one paper that has been presented, but is on bioRxiv, but is out in submission now, but which is looking at kind of a factor that we identified that's in our protocol called recreational reading. It's really just a measure of how much the kids are reading for pleasure. And we see evidence there for a kind of gene environment correlation, which seems to be contributing to the high heritability of the crystallized intelligence measure. And this has been suggested by a number of behavioral geneticists, a very well-known behavioral geneticist, and modeled actually as a likely factor contributing to increased heritability of phenotypes that are actually the ones that we think of as being most dependent on culture and education, and which is kind of counter to the conventional G theory of the heritability of G and so on. So, so we're not the only people thinking this way, lots of people are, but that was one example where we saw something we thought you could most easily explain, higher heritability of our crystallized intelligence measure an association between at least this one experiential factor, recreational reading, and that higher heritability than the fluid intelligence measure. But
Great,
you know,
thank you.
it's hard to prove this stuff, and but I think we have to be trying to model those experiential and environmental factors that appear to be correlated with genotypes in our kids, and they can be for a lot of different reasons, and you know, and how that relates to their heritability, whether these effects seem to mediate some of that heritability. And I think there are ways to make progress, but it's complicated.
Great, thank you. Yes, in addition to the measurement issue of how to characterize the environment well, as you were talking about, and I think those are very important questions for the future. There's a couple of questions here if you can answer. One is from Andrew Michael. Does the DEEP platform, I think that's what it's called, allow to set up new with stars around it analysis on the cloud? Downloading the ABC data set is an issue due to its size.
Yes. Okay. So let me see. So the DEEP at the moment, I believe the DEEP, you definitely can set up a new statistical model with the DEEP on that to operate on the tabulated data in which is all available through the DEEP. There's no raw imaging data, but I think that if it hasn't gone live yet, it is about ready to go live. I have to check to be sure, but and I don't know if BJ is still here. She might actually know the answer to this question, but the pre-processed imaging data is going to be available through the DEEP as well. This is a more computationally intensive way of analyzing the data to go directly, if you're interested in the imaging data, to have actual, you know, vertex level or voxel level data particularly vertex level data in the DEEP to analyze with all the other variables. That I think is, as I say, either has just gone live recently or maybe now, but certainly is planned. So it depends on what you're asking about. Are you talking about new methods in the cloud with the raw imaging data, or are you talking about new methods in the cloud with the tabulated data? Because for sure you can compute you can even compute you know, new measures from the ones that are there and save them as your own and go back to them, or you can leave these new measures there for other people to share. So there's lots of things you can do with the DEEP to create new variables, manipulate them. As I say, you know, the R script is just right there for the default measures, and then you can just change them or completely replace them with your own R script or whatever you'd like. I hope that answers your question. I'm not sure if it does, but.
Great, Andrew, is that okay? Unless you unmute, we'll move on. Thank you very much. Alexander, there's several more messages. Alexander Franco from the CMI. If you were able to restart the ABC study today and given the lessons learned, what major changes would you do to the current study?
Oh, God. That's tough. I think that I think that we are one thing that I think that in the study we are trying, it's a struggle for us to keep up, is to use novel technologies that might allow us to, with less burden on kids and parents, collect information from them, you know, in a little bit more temporal density. And the problem has not been that we recognize the advantages of doing this, but that like developing the informatics for it has been challenging to do with the kids racing through the developmental time, you know, and no opportunity to pause and work on our methods. So I think this is something that if we were to start over again, we would really hope to be able to put these things in place earlier. So the assessment battery is a lot of questionnaires, a lot of parent report, particularly in the beginning. It's inevitable. And then we have the cognition toolbox, and we have some other performance measures. We have a lot of psychiatric diagnostic interview. We do the case ads for a comprehensive psychiatric diagnosis. We look at the CBCL, a number of these parent-reported measures of behavioral problems in kids. And so those of you who do these kinds of assessments, kids will recognize these names. So a lot of those very reliable and well-validated methods for looking at behavioral problems in kids, all parent report and some youth report. And of course, some performance measures, as I've mentioned. But I think, it is, especially when these are being asked over and over again over time, and we're having to update, what's happened recently, it can begin to feel kind of burdensome to parents. And so ways of collecting this data more efficiently, perhaps doing it with sampling on mobile apps, so that there's not this sort of relying on people for at least a year and sometimes 2 years, depending on which measure it is, to reconstruct what's happened in time. I think it's, we could do this with better technology now, and it would be more acceptable. It's also, even since we started ABCD, the number of kids who have their own mobile devices has really increased tremendously. And in the beginning, we really couldn't rely on the youth or even the families to have a cell phone. But now I think it's much easier to, you don't have to worry as much about losing people, low-income families or families from different communities by using an app, for example. So I think it's easier. And, the other thing I think we would do is maybe provide some devices to the whole. I mean, this would have been expensive and it would have been a hard sell in the beginning. But I think if I was started again, I would consider just providing devices for all the families that could be their ABCD device. It just would solve a lot of technical, especially it would have been nice when we headed into the pandemic. Boy, that was very challenging because we lost all of our face-to-face visits. Everything had to be virtualized immediately. Families were in very different circumstances with respect to what devices they had available to them. And so I think it would have been, it would have been great if we had had an ABCD supported device. But everything with 11,000 participant families, even the smallest thing you do is very expensive.
I bet. Thank you very much. We're going to continue, but Alexander Franco also shared his CMI, kind of the tool, the MindLogger, that you and many of you probably know, and it's in the chat box if people are interested in looking at this. If you have several more minutes, I'd love to ask a couple more. I think they're lately coming in now, and so I'll try to be brief, but let's see. Were the recreational reading findings on the brain and recreational reading modeled concurrently or longitudinal? It could be a brief answer, or you can say, look at the paper and we can move on as well. But if you have that, we have 3 or 4 more questions if we can cover.
The recreational reading measure that we had was a parent report of, we had a question on an activities questionnaire that said, does your child read for pleasure or read in their spare time, and if they said yes, then we asked them about how many hours per week they did that, and that was kind of our measure.
Great.
And it was really just a concurrent kind of in the last. These are mostly activities about what has happened in the last 4 months or in the last year, things like that.
Great, thank you. From Paul Sander. He says, wonderful. Have you looked at any diagnostic subgroups, for example, ADHD in correlation with imaging G and E?
I have not, and I know that there is a very active interest group within ABCD looking at ADHD. And so I'm sure there's going to be stuff coming out. And honestly, this is an open science model. The data are out there every year, and there are hundreds of investigators. There are actually 1,300. Last time I checked, I think there were over 1,300 individual authenticated users who had ABCD data, and they're in countries all over the world. And papers are coming out all the time, and it's really impossible for us to keep on track. On top of that. But there certainly are ADHD participants in the cohort, quite a lot of them, as you can imagine. And there are people who are looking at that. I just am not aware of what they're finding yet.
Yeah, I can imagine. Maki Koyama asks if between the 2-year intervals, if you're documenting particular factors that could influence development, for example, onset of illness, when they took medication, injury, significant emotional kind of disturbance, and so on. And of course, that's kind of related to the mind logger, I guess, issue that was brought up by the CMI group. But if you're, to the extent that you know.
Well, either for parents or from youth themselves. Of course, the youth are getting older now, so we're beginning to ask them more questions of themselves, but we also continue to follow up with all their medical history with their parents. So we try to fill in everything that's happened since the last time we saw them. So we have a lot of longitudinal measures like that, if that's what you mean. Of course, it's difficult for us to time all that. We don't really have in detail when those things happen. That's the issue. There are some things that we do at the 6 monthly phone interviews. We do check up on. We check for a few things. Has this happened? Has that happened? A lot of those are things like substance use, because we're obviously trying to pick up the earliest substance exposure in these kids. That is our lead institute, NIDA, and we certainly owe them that. And so we're trying to, we ask them kind of as the kids are aging and the youth themselves, what are they taking? What are they doing? Also other things about their habits and so on. So that's a 6 monthly kind of follow-up. But we do try to at least annually get these basic, what's happened in their medical history and stuff like that.
Great, thank you. One final question, if that's okay with you. So the final question is, let me see, final question here is, what is the retention rate? So specifically it says, what is the participant retention rate expected at the beginning and the one the project currently has? Is it associated with demographics?
Yeah, I think our retention rate is 98% at this point, or 90, close between 98 and 99%, I think, at this point.
Wow.
But we consider that our retention rate is the number of participants that we enrolled who have not withdrawn from the study. But obviously, we've just been through a pandemic, and it has been disruptive to our visit schedule. There's no question about it. We had to pause and virtualize all of our assessments, as many as possible. So we are trying to keep track of very carefully our missing visits. So that's another issue. These are not people who've withdrawn from our study, but they are visits that we could not manage to get in, especially in this environment. We have done pretty well. If you look at whether the things that we could do virtually or as a hybrid visit, we also have a hybrid visit format that, as became to be a little bit safe and became a little bit safer. We had mostly virtualized visits, but a few things that are hands-on that we couldn't get any other way, some anthropometrics and things like that we do hands-on with the kids. We have, and the imaging, of course. We began to bring people in for a shorter on-site visit as a hybrid visit, just for that part. And of course, now we're beginning to see people almost opening up completely, but with a lot of social distancing restrictions mostly in place. So, we have this mix of different kinds of visits, especially during the pandemic period. And then we have some missing visits, and we obviously have a little bit of an elevation in our missing visits relative to visits that were skipped by families before. And so we are in the process of really trying to take stock. How many of our families were displaced, lost their homes, no longer are available to us? Can we find them? These are families who have not withdrawn, but we know that the pandemic has been very hard. We have a lot of low-income families in our study compared to most studies. And we also have a lot of families who are from different communities who have been impacted particularly hard, extremely hard by this pandemic. And so we're really trying to take stock right now. But we have still a very small percentage of families who withdrawn from ABCD. And we try to
That's
get a lot of
impressive.
contact information. So we get, at every time we try to get contact information for all their associates and so on, so that we can find them when we come out of this.
Great. That's wonderful. That's amazing. Well, thank you very much. And I will share the positive comments and the compliments that we received of your talk, Terry, afterwards. But thank you very much, everyone, for joining us. Have a wonderful summer. And Terry, thank you for your time today and sharing your wisdom and your knowledge and your studies. Thank you.
Well, thank you so much for your attention. It's been great and very a little humbling to see all these wonderful faces and names out there. And thanks for your interest.
Thank you very much. Bye, everyone.